In the previous class, we have studied about some of the applications of the biotechnology in the field of agriculture. We already done in the previous class, and then yesterday's class. Of course, yesterday were doing the this thing. Today we'll study about the application of biotechnology in uh, therapeutics. That is the treatment. Okay, so we got insulin and then we got ADA. Okay, so let us see. Let me share the book and let me pin it up. Okay, so we got here the management of adult onset diabetes. Adult onset by diabetes का मतलब क्या हुआ? That is the diabetes which is caused in the later stage of the life, not at the birth. The diseases which are there since birth are called congenital disease. So of course there are cases in which the diabetes are started so early in the ages, but most of the cases the diabetes is caused in the later stage, that is after one is reaching to the adulthood. So we are talking about the management of adult onset diabetes is possible by taking insulin at the regular interval of time. So what is the simple way of treating this disease is take the insulin. Insulin is available in the market. It is available in the form of injections, and one has to take those injections at the regular interval. And depending on the test and all, one has to keep on doing the test. Okay. Now there are some people who are without testing; they are simply taking insulin. So they don't even come to know that how much insulin is required. And many a time the thing is that whatever amount of required, bare minimum required of glucose is there, that is we have studied, that is from the 80 milligram per 100 ml blood to 120 milligram per 100 ml blood. So if it goes below 80 milligram per 100 ml of blood, that is the sugar level, and it remains there for sufficiently long time, then the person may get fainted. The person may even go into coma. So it is very necessary that the patient who are taking insulin, they should regularly get themselves checked. Otherwise, it will lead to the severe problem. Okay, so now talk about this thing, that is how what would be the diabetic patient do if there is not human insulin available. If we discuss, so we got only two ways: either limit the consumption of sugar. Now sugar is not in the form of sugar only. Whatever are what you call staple diet is, or staple diet, may it be wheat or may it be rice or may it be maize. So they contain quite a good amount of starch. Maize and rice. Rice to contain maximum starch. Sorry, not maize, wheat. Wheat contain again quite a good amount of starch and a little amount of protein and some minerals. And the third one is maize. Maize got a lot more quantity of protein than the starch. Okay, so whenever these food items are consumed plus other food items are consumed, definitely they have got some amount of starch and some amount of other form of carbohydrates, and on digestion they all change into sugars, glucose. So that you simply don't think that is if I don't take glucose, it will not lead to the problem. Whether one is having rice or whether one is having wheat, chapatis, or whether one is having idlis or dosas or uh, whatever, any item made up of sabudana or soya bean even, or any other, sago even, or any other thing, they are mainly containing starch. So starch on digestion will change to sugar only, and sugar will have this type of problem only. The excess of sugar if there, it leading to diabetes. So if we discuss that this will be soon when will be soon realize that we would have to isolate and use insulin from some other animal if there is no insulin available. If our body is not able to make insulin enough, then what is the possibility? First, to limit the amount of glucose or carbohydrate intake. That will again lead to other problem which we already discussed in the previous class, that is when I was teaching about phenylketonuria. I said if we take more of protein, then it will produce ketone bodies, and that will again lead to the another problem. So more of urea and more of nitrogenous waste will produce, and that will create another situation. So we are not actually meant for those things. Only the true predators are there which are consuming only protein, and they have got very little carbohydrate. We got more of carbohydrate and some amount of protein, because we got thoroughly in all the parts of our digestive system, protein digesting enzymes are there. That supports that we are actually meant to eat quite a good amount of protein, but not solely on protein. Okay, so then what we can do is we can reduce the intake of carbohydrate depending on the body's actual require. That can be done. That is a way of monitoring. And otherwise what we have to do is we have to take insulin from other sources. Now what are the other sources? When as far as the insulin was not being able to produce by biotechnological experiments as we are studying today, till then we were depending on the insulin from the other sources like animals, and that it used to lead to a number of problems. Okay? Why? Because the insulin which is made in the other animals may not be suitable for us, because they may have some other type of substances also which may lead to the immunological immunity type of problem. That is, they used to lead to some of the what do you call allergy or some other type of immune response. So that is why it was not good for us to completely depend on the animal insulin. Moreover, animal insulin itself was available in a very small quantity, because if suppose one goat is slaughtered, there may be hardly 1 ml, 2 ml, 3 ml or 5 ml insulin will get from its pancreas. 5 ml insulin can be used for how many people? And how many animals are slaughtered every day? So how much insulin? So that insulin requirement from the other animals either would have not been sufficed, nor it used to lead to the immune responses and other type of problems. And so that was not a very good alternative. Okay. Now imagine if the bacterium were available to that could make human insulin. So the scene is very different, because we can produce any number of bacteria and we can easily culture them with the bare minimum type of conditions, and we can get any amount of insulin. And that also what we are talking human insulin. Human insulin means that is those immune response which used to be caused because of the intake of insulin from some other sources, that would have also been reduced greatly, or rather that would have never been there. Suddenly the whole process becomes so simple, and you can easily grow large quantities of bacteria that can make much insulin as you need. Think about whether the insulin can be orally administered to the diabetic people or not. Now see, insulin is actually polypeptide chain. There are two polypeptide chain found in the insulin, polypeptide chain A and polypeptide chain B. And we got number of protein digesting enzyme in our stomach, pepsin, then duodenum, trypsin, and then as it goes into the small intestine, we got amylopeptidases and chymotrypsinogen and that and this, and there are so many type of protein digesting enzyme. So it would have been digested, it would have been broken into the amino acid. So then its function would have not been, because the insulin cannot be directly absorbed in the mouth or in the alimentic in the pharynx region, and no sooner it will reach to the stomach, it will be denatured. So that is why of course there are methods and methods like in the form of tablets or capsules or some other form they should be taken in, so that but it will remain inactive in the stomach and the duodenum, even in the small intestine, and by the time it reaches to the large parts of the large intestine, small intestine, where they can be absorbed. Till then, if they can reduce, and then again our body cannot take the polypeptides in, so they have to be broken down into the amino acid, and if it is broken into the amino acid, it is no more insulin. So that is not a good solution. Now, insulin used by the diabetes was earlier extracted from the pancreas of slaughtered cattle and pigs. Cattle and pigs, these books are actually churowed, copied from the foreign books. Okay? Where in India cattle are consumed, where in India pigs. Pigs are hardly in certain states, and cattle are also only in certain states. We are a country where cattle are what you call worshipped. So these type of things you should not see also. I don't know who writes these books and who what to say. Chaliye, now, insulin from an animal source though cause some patient to develop allergy and other type of reaction to the foreign protein. Insulin consists of two short. ये paragraph खत्म हो गया, नया paragraph शुरू होना चाहिए. Now क्या? That insulin consists of two short polypeptide chain. Here you can see a blue color chain is there and a green color chain is there. The blue colored chain is polypeptide A and green one is polypeptide B. Then there is a gray color chain also. The gray is polypeptide C. In human being, the insulin is secreted in the form of proinsulin in this form. Here to here is polypeptide A. I'll simply write A. Here to here is polypeptide B. And from here to here, the polypeptide C. C is not needed at all. पर ये है, पता नहीं क्यों है. So the insulin is secreted in the form of proinsulin, and there are two disulfide bonds here, and one disulfide bond is here. So when the proinsulin matures and change into insulin, this is the structure. This has to be removed. So the body has mechanism that the proinsulin is changed into insulin and this structure is formed. So the thing was very easy. What we had to do is we had to produce two polypeptide chains separately, study the gene where they are located. By human genome study, we have already come to know that where the insulin A gene is there, the polypeptide A gene is there, for insulin polypeptide B gene is there. Those genes have been cut, extracted, added to the bacteria or yeast, whatever culture you want to use, and then these polypeptides were extracted from there. Okay, downstream process. But again, there was a big challenge. The two polypeptide chains are separate. They have to be joined together by the disulfide bonds. And we were not able to do it for quite some time. C polypeptide, of course, we need not to make. Why we have to make? Okay, in the naturally occurring polypeptide proinsulin, the ACB is there, and where the C has to remove an A and B are already joined there. Okay, but when we are making biotechnologically this, so we have made the separate polypeptide A chain, separately polypeptide B chain, and they were joined together. And this joining was performed way back in 1983 by an American company, Eli Lilly. वैसे तो बुक के अंदर ऐसी कंपनीज के नाम नहीं लिखे रहते हैं, but since it is a discovery, so it is mentioned here. So in 1983, Eli Lilly, an American company, prepared two DNA sequences. That is, two DNA sequences. They are cut from the in the human chromosomes, and they were entered into the or added to the bacteria, and two polypeptide A and polypeptide B corresponding polypeptide chains were formed, and then they were artificially joined by the disulfide bond. So it was just exactly similar to that of the human insulin. So this will not create any type of allergic response. This will not create any type of immunological problem. So this was very safe, and this is how today we are making insulin. So now the thing is that insulin is available in plenty. कितना चाहिए ले लो. Why? Because bacteria are involved. Okay, and the whole process is very easy. Now we need not to slaughter any animal. Now we need not to extract the insulin from the animals. Now we need not to fill them, store them, keep them safe and all. And in spite of that, when somebody takes, the one has to see that what type of allergy, some allergic response are there or not there and all. So all those things are now become very safe for human being. So thanks to the biotechnology, and as you know, just for the general knowledge, India is the capital of diabetes in the world, the maximum patient of the diabetes are found in our country. Okay. चलिए another this thing we'll study, that is called gene therapy. If you study that insulin case, diabetes case or adult onset diabetes case, that means at the time of the birth the gene was okay, but later on the gene's functioning has been affected, maybe some reason or other, maybe mutation, maybe some other type of problems were there. There's some inability of the body failed to produce enough of insulin. But the gene therapy is a different picture. It is a genetic disease, which is a person is born with that type of problem. Okay. So now what we have to do is either we have to correct the gene. That means like you got a TV set, and in the TV set, suppose one particular transformer is spoiled, or one IC is spoiled, or one speaker is making funny noise. So what you can do is when you call a mechanic just see that which part is not good, and that is being replaced and new one is put. If that kind of thing can be done in the human beings, can we remove the defective gene and put a correct gene? Or can we add a functional gene in place of the non-functional gene, or let the non-functional gene be there? We can bypass its function and we can add another functional gene. So that was the strategy in the gene therapy. So in gene therapy, what we are doing, when a person is failed to produce the particular enzyme, ADA enzyme, that is adenosine deaminase enzyme, what we are doing, we have seen that which particular gene makes ADA, that gene is being cut from somewhere, that is cultured, and that gene is, that is culture means that is cloned, and then those genes are added into the WBCs, lymphocytes, and those lymphocytes are sent back to the child. Now why we are adding into the lymphocyte, why not in some other part? Because lymphocytes are only responsible for immunity, and these genes are functioning in the lymphocyte only. But the problem is again that the lymphocyte is a short lifespan. They can live only from 10 to 15 days. So that means again you will have to keep changing those lymphocytes. Let us see how this thing. Okay. If a person is born with the hereditary disease, that means congenital, पैदाइशी है बीमारी. It is not like that or adult onset. Can be corrective therapy be taken? Can a corrective therapy be taken for such a disease? Yes, of course. कैसे? Gene therapy is an attempt to do that. Gene therapy is the collection of method that allows the correction of a gene defect that has been diagnosed in a child or in an embryo. Now, if you remember Alec Jeffrey. Who was Alec Jeffrey? You can speak. Who was Alec Jeffrey?
So,
what was his call?
So that parentity one.
क्या कहते हैं उसको? DNA fingerprinting.
Yes, sir. DNA fingerprinting.
Okay. So DNA fingerprinting. Alec Jeffrey was not searching for DNA fingerprinting. देखिए ना, मतलब दुनिया में पता नहीं क्या क्या होता है. वो अंधे के हाथ बटेर लगना इसको कहते हैं. अंधे के हाथ बटेर लगना. जॉन ग्रेगर मेंडल वो स्टडिंग मैथमेटिक्स, स्टैटिस्टिक्स पढ़ रहा था, और क्या लग गई बटेर उसके हाथ में? हेरिडिटी, है ना. This person Alec Jeffrey also, he was studying this type of things, that if one of the parent is having any genetic disorder, what is the probability that we have come to know by the pedigree analysis, that whether the gene is dominant or recessive, whether it is what you call autosomal defect or it is chromosomal defect. So what will be the probability that we come to know? So if we see that there is any hope of getting a infected child, matlab the diseased child, or a disordered child, or a normal child, if there is a possibility of getting normal child, तो आप क्या कर सकते हैं? We cannot design an experiment कि we will help only the healthy that is sperms and egg to fertilize. We cannot see that what is there in the chromosome, that is the sperms chromosomes, and what are there in the X chromosome, and that is taking place inside. And so far we are not able to make such mechanism to separate only that. जिस दिन वो आ जाएगा ना, समझ लीजिए सब गड़बड़ हो जाएगा. वैसे साइंस ने गड़बड़ी करे. We see only the positive part of the science. There are so many negatives are going on. So अगर ऐसा हो जाने लग जाएगा ना कि we can separate that what desired chromosome can be, the sperm can be taken and what desired egg can be taken, they can be fertilized. तो कौन फिर X chromosome को ले जाएगा? सब Y chromosome को X से fertilize करके लड़के पैदा कर लेंगे. Country like India, where the male and female are having so much of differences in their culture or in their society. तो वो छोड़िए, अब बच्चा तो पैदा हो गया. पैदा होने के पहले when a child is formed in the embryo, in the uterus, by amniocentesis we can come to know whether the person is having normal gene or defective. And if the defective gene is there, we have got two options: either ask the parent to continue with that or not to continue with that. Not to continue, the problem khatam ho jayegi, but that is having certain other problems like that socially, religiously, ethically, and other way it is not been accepted. Now, agar usko sansar mein lana hi hai, so that the child will have this defect throughout their life. So now, what is the solution? What is the solution? तो solution क्या है हमारे पास? उसका एक ही solution है. क्या कि embryonic stage में ही हम उसका gene replace कर दें. Let us replace the gene in the embryonic stage. Now it is a very tedious process. The child is in a fetus, and in the fetal stage only you want to replace the gene. So that will be a thing. So देखिए क्या लिखा है? Correction of genetic defect involve delivery of normal gene into the individual of or embryo to take over the function of compensate for the non-functional gene. The first, now that is not in our course. No, that is again if you study in the higher classes you will study. Okay, now what other method is there? A child is already born and the child, a girl, suppose this is an example given for the girl. So a girl is having this defect. She already born and she was already reached at the age of 4 years. And when it has been found that the girl is all the time suffering from disease because her immunity is very low. So on the study they came to know that she is not producing enough of adenosine deaminase enzyme. So what is the solution? What is the solution is either you give adenosine deaminase enzyme from outs, just like insulin which we are giving. That means the girl will have to be poked every day. Just insulin ke liye, the person who is suffering from diabetes, they have to take insulin every day, sometimes twice a day. Okay, so sometimes before every meal. So it depends on the quantum of disease. So that would have been a problem as such a small girl of 4 years. Every day she has been poked. It used to be a painful experience. And this is only an example. There are many who are born with this disease, so they have to be given ADA right from the childhood, right from 6 months age or right from 3 months age. So that will be a very painful experience. So देखिए, the enzyme is crucial for immune system. क्या हुआ उस बच्चे के अंदर? A clinical gene therapy was given in 1900 to a 4-year-old girl with the adenosine deaminase deficiency. And the enzyme is crucial for immune system to function. So immune system क्या है? Immunity नहीं बन रही है. How the immune system will work? The disorder is caused due to the deletion of a particular gene, that is adenosine deaminase gene for this. Okay, in some children the ADA deficiency can be cured by bone marrow transplantation. There are a number of options available. So option number 1, just either in the embryonic stage only or no sooner the child is born, immediately all the bone marrow, the defective bone marrow will be removed and a fresh bone marrow which is suitable for the child according to the tissue match and all, that will be given. So then it may be possible that the person's bone marrow is correct, then it will make the right type of RBC, WBC and platelets, and WBC will have lymphocyte and lymphocyte will be exactly similar in function, so that problem can be got over. But this is a very risky thing and that has to be done as early as possible. So bone marrow transplant. In other, it can be treated by enzyme replacement therapy, which is same as insulin layer. That is, every day you have to give the enzyme from outside. With the functional ADA is given to the patient by injection. But the problem with both these approaches that they are not completely curative. Okay, so now as a first step toward the gene therapy, lymphocyte from the blood of the patient is grown in the culture outside. So first what we have done, this is the girl, her lymphocytes are taken out. And these lymphocytes, whatever are the lymphocytes, these lymphocytes, they are given the healthy genes from outsource. Okay, so now this will be known as complementary DNA, that is cDNA, and then these will be reintroduced into her. So since the healthy genes are there, it will make enough of ADA for a certain period of time, maybe up to 4 days, 5 days to 15 days, depending on the lifespan of this lymphocytes. Okay, and so this will be helpful. So and functional ADA, that is the cDNA, using the retroviral vector. Now, what retroviral vector? We already studied that there are some viruses which are RNA bearing, and those virus have an ability to enter into the human. So, वो अपना gene, अपना genome, they can insert into the human body. So, with that, we attach this particular segment of RNA which is making what you call ADA. If that is added, so it will enter in the body and it will be attached and it will start functioning. So is then introduced into the lymphocyte, and then they are subsequently the patient requires the periodic infusion with genetically engineered lymphocytes. So picture कैसे बदल गई कि earlier we had to give every day in this ADA. Now we have to give it periodically, maybe after every 10 days or maybe after every 12 days or 15 days. However, gene isolate from the marrow cell producing ADA is introduced into the cell at early embryonic stages. It could be a permanent cure. Okay, so this is called gene therapy. Gene therapy and that intake of what you call insulin, they are two different things. Okay, intake of insulin that is a lifelong process, and where the insulin is produced outside and then insulin is given into the body. Here we are not giving enzyme from outside. That is all one of the way of treatment. But we are not in this particular case, gene therapy. What we are giving, we are taking the cells, that is a lymphocyte from the patients out. Those lymphocytes are cultured, and in which the healthy gene, normal gene, functional gene is added, and then with the what you call this gene will be added by the virus, okay, retrovirus, and then such DNA containing lymphocyte will be added back into the person. So this will be functional. Okay, so that's all for today. Now tomorrow we'll study molecular diagnosis, and tomorrow is Friday. I thought to finish the chapter, but it's not possible. Chaliye. Thank you, Jalan.
Thank you, sir.
Welcome. Stop sharing.